Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. What I have for y'all today is mostly a study guide. We've been talking about a lot of complicated stuff, replication, transcription, translation. And so what I wanted to do today is just run through the study guide with y'all. We can kind of work through this together, and then that way you can take this home, look over it on your own time, and you'll be able to ace that test that you got coming up. So without any further ado, let's go ahead and hop into it and see what we got going on today. All right. So we're going to go ahead and get started. Number one here says, fill in the blanks below with the correct information. It says DNA goes into what, and that goes into protein. What we should write here is RNA. Remember that this is the central dogma of biology. This is transcription and translation. That first arrow represents transcription, going from DNA to RNA, and then that second arrow represents translation, going from RNA to proteins. Okay? So number two, this says the first process in protein synthesis is known as what? I just said it. This should be transcription. All right, and then what is the second process in protein synthesis? That's translation. Great. All right, let's keep it going. 4 says, match the scientists with their contributions to the discovery of DNA. So we've got 4 scientists or 4 scientist pairs, and then we need to match them up with what they kind of figured out. So let's look at this first one. It says credited for discovering the structure of DNA. So this is probably the most important one. It's the one that's most likely to pop up on your test or on the state test. Who gets credit for discovering the structure of DNA? That's Watson and Crick. So really try to remember their names, and that's going to be letter C. Okay, the second one says, we're able to take incredibly detailed pictures of DNA using X-ray crystallography. This is going to be our other pairing here, D. That's Rosalind Franklin and Maurice Wilkins. They had a technique that they were able to take pictures of DNA. That process was called X-ray crystallography. Here's the next one. Who was an American studying proteins as the genetic material? This guy's name was Linus Pauling. He was kind of a really big deal back in the 50s and 60s, but he didn't really have the right idea when it came to DNA. He was looking at proteins and not DNA for the genetic material. And then the last one here, found that the amount of adenine equals the amount of thymine, and the amount of cytosine equals the amount of guanine. That's this guy known as Chargaff. These represents Chargaff's rules. A and T go together, C and G go together. At the time, he didn't really know what that meant, but it ended up being a really big deal for these other scientists studying DNA. So let's keep it going. This says, use the diagram on the right to answer the questions below. So what we're doing here, this is a DNA molecule, and it's our job to figure out what these different numbers on here represent. Okay, so 5 right here says the green bases. Okay, well, how could we figure out what this green base is? It's not labeled as anything, but I do see that the base that it's pairing up with is known as thymine. So what base pairs with thymine? Well, that's adenine, right? So this one should be B. And again, how can you remember that adenine and thymine go together? Some people think of the word apple tree. I just like to think of the word at, A and T. And that's how I remember that these go together. Okay, let's try the next one. 6 says the red bases are. So here's 6, here's that red base. It's not labeled as anything, but we can see that it's pairing with guanine. So what nitrogen base pairs with guanine? That's going to be C, cytosine. And for this, some people think car garage. That's a way to remember that C and G go together. All right, let's keep this going. 7 says the sides of DNA are. So all of this makes up the side of DNA. And we call this the backbone. What makes up the backbone of DNA and RNA? Well, this is a sugar phosphate sequence. So you can see here that all these yellow things have the letter P in them. That's the phosphate. And these shapes, the pentagons here, represent the sugar. So it's a sugar phosphate backbone. So the sides of DNA are made up of A, phosphate and sugar. All right, here's the next one, the bonds between the bases. So what it's asking here is these bonds here, the little dotted lines. What type of bonds are those? Well, those are hydrogen bonds. Hydrogen bonds are not very strong bonds. They're very easy to break. And that's good because we have to split this molecule down the middle when we replicate it. So hydrogen bonds, they're strong enough to hold it together. They're weak enough to break apart when we need it to. All right, let's keep it going. All right, so we'll go to number 9 next. This says the two sides of the DNA molecule are. Well, I'll draw it over here and then maybe we can find the right answer. So this would be the arrangement of those DNA molecules. They're side by side, but they're actually going in opposite directions. And so what we call this is anti-parallel, and that they're running in opposite directions. So they're parallel because they run side by side and they never cross over. But they're anti-parallel because one's going in one direction and one's going in the opposite direction. Great. All right, 10. The small fragments being made on the lagging strand are known as what? So these are known as Okazaki fragments. This was discovered by a pair of scientists who were married at the time, and their last name was Okazaki. And so they were researching this at the time. They found how these little fragments had to be joined together to make a long strand on the lagging strand. And so in honor of the couple, they named them Okazaki fragments. Okay? I'm glad they were married. Imagine if there was 2 long names you had to remember. All right, so 11, this says for each DNA molecule being made, let's look at this. So the term I want us to know here, we're going to go with letter A. One of the strands is new and one of the strands is old. We call this being a semi-conservative. So let's go back to my drawing here. So imagine this is what you're starting with, okay? If we were to make new strands of DNA, it would kind of look like this. So what's going on here is the blue strands are old, right? They came from our original strand. The red ones are new, and so we call this semi-conservative. One of the strands is new. This red one would be new, and one of the strands is old. That blue one is old. It came from the original DNA molecule. So that's called being semi-conservative. All right, let's go to 12. Match the function with the correct enzyme. Okay, so looking at these, number 12, which one of these hacks or unzips the DNA is going to break those hydrogen bonds? Well, I'd like you to remember that helicase hacks. They both start with the letter H, and to hack something means to cut it in half. Okay? The next one, primes the new DNA strand. Well, again, looking at the name, there's kind of a hint here. What primes the new DNA strand? Primase. They kind of sound alike, right? What it does is it lays down an RNA primer, and that tells DNA where to start replicating. Okay? Which one pastes new nucleotides? Well, that's going to be our polymerase, so A. When we're replicating DNA, we use DNA polymerase. When we're transcribing and making that mRNA, we use RNA polymerase. All right, and then which of these links the nucleotides together? That's going to be B for ligase, and just think links, ligase, both starts with the letter L. Okay? All right, 13. Replication, transcription, and translation all have the same 3 steps. What are they? So as this unit went on, replication, transcription, translation, they all happen in 3 steps, and for all 3 of them, the 3 steps were the same. So what were those words that we were saying? Well, the beginning of replication, transcription, and translation is known as initiation. So that's one of them, okay? Once you initiated it, you would actually start building the DNA molecule or the mRNA molecule or the polypeptide, and that was known as elongation. That's the second step. And then the last step is termination, and to terminate something means to end it. So all 3 of those processes had the same 3 steps, and those are those 3 steps. All right, let's look at 14. This says, how are each strand of the DNA molecule replicated? Okay, so we have a leading strand and a lagging strand. What I'd like for you to remember, let's go with B for this one. The leading strand is the one that's easy to make. We say that it's made continuously. Okay, it's made all at once. The lagging strand is the harder one to copy, and we say that that's non-continuous. What happens then is we make a lot of little fragments. Those are called Okazaki fragments, and then ligase has to come back and link all of them together to make a long strand. So the lagging strand is non-continuous and the leading strand is continuous. Great job, y'all. Let's keep it going. Okay, 15 says, put the following events in order. Use the numbers 1 through 4 in the blanks below. Okay, so these are the steps of DNA replication. When we're replicating DNA, the first thing we have to do is open the DNA molecule up. So this is going to be helicase unzips the DNA molecule. That's the first thing that has to happen, and let's remember that helicase hacks. Okay? The next thing that happens, now that the DNA molecule is open, those nitrogen bases are exposed, this DNA primase is going to lay down an RNA primer. Okay, so that's number 2. Then, once we have the primer, just think of the primer like a starting point. Once we have a starting point or the starting line laid down, the next step is DNA polymerase is going to start adding nucleotides to the growing strand. And then the last thing that has to happen is Okazaki fragments are linked together using ligase. Okay? All right, let's come down here to 16. So this is very similar to our Venn diagram that we did a while back. What we're going to do here, in each of these blanks, we're going to use the letter D if only DNA does it. We're going to use the letter R if only RNA does it. And if both of them does this or has this, we'll use the letter B. Okay, so your options for answers are D, R, or B. So let's see what we got going on here. So which of these is in the nucleus? That should be B. DNA is always in the nucleus, and you can find RNA in the nucleus. So that's both. All right, which one has 3 types? Okay, let's write an R for this one. RNA has 3 types. Okay, which of these has thymine? Okay, thymine can only be found in DNA. Okay, which of these is a nucleic acid? Well, deoxyribonucleic acid and ribonucleic acid are both nucleic acids. So let's go with a B for that one. All right, which one of these has a sugar phosphate backbone? That's going to be B as well. They both have that sugar phosphate backbone. Which of these is genetic material? That's going to be a B as well. They are both genetic material. Okay, which one has uracil? Well, remember that DNA has thymine and that RNA has uracil. So let's put an R there. All right, which of these is single-stranded? That's RNA. Which one is made up of nucleotides? Well, remember that they're both nucleic acids, and all nucleic acids are made up of nucleotides. So they are both made up of nucleotides. All right, which of these has sugar as deoxyribose as the sugar? Well, that's DNA. Which one is used in replication? Well, that's DNA. I guess you might even be able to put B here, though, because we do use an RNA primer. So let's switch this one over to B. I had this one as D in my key, but thinking about it now, you do lay down an RNA primer, so you really use both of them. All right, which one of these is used in translation? Okay, so if you think back to our process, translation happens in the cytoplasm, and so DNA is actually not part of this process, so it's just an R. Okay, which one of these has ribose as the sugar? Well, that's RNA. What has cytosine, guanine, and adenine? That's going to be both. Those are the 3 nitrogen bases that they both share. Okay, which one of these is in the cytoplasm? That's only RNA. Which one of these is double-stranded? That's DNA. Which one is used in transcription? That's both. You use DNA as a template and you make RNA out of it, so you use both in transcription. And which one of these is needed in protein synthesis? They're both needed in this process because you transcribe the DNA first and then you translate it into proteins. Great. All right, a couple more. Y'all hang in there with me. All right, here's 17. For this, we're going to get some copy transcribing. So this line right here is DNA, and our job is to transcribe it into mRNA. Remember, RNA does not have the letter T, it has U's instead. So we're never going to see a T on this line. So what I have is UAG, CUU, AAU, ACG, and then lastly UCC. Okay? This says, now that you have the mRNA, find the anticodons that are complementary. So this is messenger RNA. This question is asking us to find the tRNA that is complementary. What we're going to do here is we use this line, and we're just going to use those complementary base pairs to find the anticodons. So for this one, so those are the anticodons that are complementary. Again, a little cheat for this is that it's going to look just like our original DNA sequence, except for we don't have any T's in it. So anytime I saw a T, I just wrote a U instead. Everything else was exactly the same. All right, so 19 said, would question 17, is this transcription or translation? Well, we went from DNA to RNA. Going from DNA to RNA is transcription, okay? And then 18 says, 20 says, question 18 would be considered, well, this is translation, because translations is when we start using that tRNA out in the cytoplasm. All right, y'all, great job. Here's the last question I have for us. It's this big picture that we've looked at, and our job is to label it, okay? Some of these can be tricky, so I gave y'all little hints down there at the bottom. If you need those, refer to them, and that'll be a little hint to make this easier. What I want to do now is just kind of review this with y'all. All right, so number one, this is asking for what is this whole picture showing us? What process is this? Well, this is translation. Okay, we're out in the cytoplasm and we're making proteins. All right, and then number two says, what is this long line here? What do we call this long line? Sometimes you might see it called a protein, but actually a more accurate name at this point would be a polypeptide. And why do we call it a polypeptide? Well, let's look at 3. It's pointing to the bonds in between each amino acid. These little black lines between the circles, what are those called? Well, those are called peptide bonds. And because there's a lot of peptide bonds in this molecule, we call it a polypeptide. Okay? Great. All right, so number 4 is pointing to this. This is a organelle. What type of organelle is this? This is a ribosome. What are ribosomes made up of? They are made up of rRNA. So that's what we should put in this blank. All right, so that's 4. Next up is 5. 5 is pointing to these 3 nitrogen bases on this molecule. What are these? Well, these are called anticodons. Remember that tRNA has anticodons in it, and it's 3 nitrogen bases. Anticodons are complementary with 6. What do we call 3 nitrogen bases on the mRNA? These are just called codons. And codons code for amino acids, and we use anticodons to make sure we're pairing them up correctly. All right, 7 is pointing to this little circle up top. What does tRNA transfer over to the ribosome? Well, each little circle here is an amino acid. Okay? And then 8 is asking, what is the name of this structure or this molecule? What do we call this? This is tRNA. Remember that tRNA has anticodons on the bottom and amino acids up top. All right? And then 9 is asking, what is this long black line? Well, we made this in the nucleus, and this is mRNA. So y'all, thank you so much for following along. This stuff is very confusing, but y'all are very, very smart. And if you practice, you can figure all this stuff out and ace your test. So your teacher has some other work for you to practice on. Make sure to give that your best effort, and I hope everybody has a great day.